This is a review video for Biology 12 synapses. The synapse is how a neuron passes the action potential from one neuron to another. If we have an action potential, we have a sensor being stimulated, and the action potential travels down a neuron through a wave of depolarization all the way down, and then it gets to the terminal branches of the axon here, and it must jump from one axon to a dendrite, and then continue to do that through another neuron, and then do that again in this model of a reflex arc. Neurons achieve this effect by releasing chemicals or neurotransmitters on a receiving cell. That will happen on the dendrite or cell body of another neuron, or on an effector, muscle or gland cell. In this case, it's on the stomach muscle. The synapse then occurs where the terminal branches of the axons meet a dendrite or cell body of the next neuron. If we take this area here and we expand it, we can see that these are axons terminating in terminal branches here and meeting another dendrite. Expand that section up, and here we have the axons terminating the terminal branches, and here's the axon, and then the other side is diagrammed, the membrane of the dendrite, and then that signal would be passed on. Now, in order to do that, we're going to take one of these sections and blow it up and explain what's going on. This is a blow up of a synapse. These little black dots will represent the neurotransmitters. These circular items with the neurotransmitters inside are vesicles. This area here is the axon bulb. Of course, this all being the axon. There's calcium +2 ions on the outside of the axon. That's a cell. There's some organelles on the inside, mitochondria in this case. The membrane of the axon in between this gap is known as the presynaptic membrane. The membrane on the dendrite side is known as the postsynaptic membrane. The intervening space between the axon and the dendrite is known as the synaptic cleft. And on the opposite side of the axon and the dendrite side, we can see that there are receptor sites for the neurotransmitters. Okay, well, let's do an overview of the action of events that are going to occur. First action of event, an action potential comes down the axon to the terminal branch. At that point, the axon membrane becomes permeable to calcium. Woohoo! Calcium, calcium! The calcium then enters the axon bulb. Whoosh, 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 whoosh. The influx of calcium then causes microfilaments to pull the vesicles to the presynaptic membrane. So microfilaments start pulling all of these vesicles to the presynaptic membrane. And now the vesicles will merge with the membrane, and through a process of exocytosis, the neurotransmitters are discharged into the synaptic cleft. Neurotransmitters diffuse in to the synaptic cleft, and as the neurotransmitters diffuse across, they will bind with the receptor sites. Now, one point: when the receptors bind with the neurotransmitters, they bind in what's called a lock and key manner, similar to what we discussed earlier with enzymes. They fit each other exactly, hence like a lock into a key. If you're the wrong neurotransmitter, you won't fit into that same receptor. Now, these neurotransmitters can have one of two effects. They can either excite or inhibit the target. If excitation occurs, then the postsynaptic membrane, now that's this membrane here on the dendrite side, the membrane potential will decrease. So its voltage is going to start to approach the plus 40 millivolts required for an action potential. And so the actual potential energy will decrease. The sodium channels will be opening. The inside is going to change from negative to positive. If the inside becomes positive enough, it meets the threshold amount, then it will generate a nerve impulse, and then that nerve impulse will continue on down the dendrite to the cell body and along the axon of this neuron. So excitation. Now the other possibility is inhibition. In inhibition, the postsynaptic membrane, again this membrane here, the potential increases. Instead of being at negative 65 millivolts resting potential, maybe it'll go down to minus 70, minus 75, minus 80. So the potential difference that it has to reach the threshold becomes greater. The inside will become more negative. There's a greater difference between the negative and positive of the inside and outside. This decreases the chance of a nerve impulse. You could have more than one type of neurotransmitter being released. The neuron we're talking about could be receiving signals from more than one neuron, and so at the same time that you're getting excitatory signals from one, you could be getting inhibitory signals from another. If it reaches the threshold, then it will fire a nerve impulse. If it doesn't, then it will not fire. A reminder, this is the all or none response. But let's pretend for the sake of our membrane, they reach the other side, they bound to the receptors, and then they did reach the threshold, and now another nerve impulse takes off, depolarizing the membrane and going on. Now, once that has happened, we have neurotransmitter. Hello, I'm a neurotransmitter in the membrane. That neurotransmitter is just sitting around, and it will continue to excite over time unless we can get rid of it. Now, how to remove this neurotransmitter? Well, one method is through the use of an enzyme that will inactivate the neurotransmitter. These speared and axed gentlemen here are representing an example one called acetylcholinesterase, which is an enzyme that breaks down acetylcholine, a neurotransmitter in the parasympathetic nervous system. So they would come in, chop, 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 and break down all of the neurotransmitter. And that way they would stop doing the response, and very quickly the signal would have been generated, and now their job is done, so they would be stopped. Another method is that the synaptic ending can then absorb the neurotransmitter, and they can just repackage it or then break it down inside of it. So an enzyme can be released into the synaptic cleft, like acetylcholinesterase and these axed and speared carrying gentlemen, and chop it up. Or it can be reabsorbed and then either chopped up or just simply repackaged. We need short neurotransmitter lifespans to prevent continuous stimulation or continuous inhibition. Once they've done their job, they need to be gotten rid of. For those of you who are away from class and just need to also make sure that they have the notes complete, here are the notes to double check. And that is the review video for the Synapse for May 26, Biology 12.